ಶುರು ಮಾಡೋದ ಮೇಡಮ್?
Yes, sir. Definitely.
So good afternoon, ma'am.
Good afternoon, sir.
So good afternoon, everybody. So ಇವತ್ತಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಶ್ರೀಮತಿ ಲಕ್ಷ್ಮೀ ಮೂರ್ತಿ ರವರು ಕಿಡ್ನಿ ಕಾರ್ಯಕ್ಷಮತೆ ಮತ್ತು ಅದರ ತೊಂದರೆಗಳು, ಅದರಿಂದ ಆಗುವ ಪರಿಹಾರಗಳು ಎಂಬ ವಿಷಯದ ಮೇಲೆ ಉಪನ್ಯಾಸ ನೀಡಲಿದ್ದು, ಸೊ ಅವರನ್ನು ಅಭೂತಪೂರ್ಣವಾಗಿ ನಮ್ಮ ಅಕಾಡೆಮಿಗೆ ಸ್ವಾಗತಿಸುತ್ತಾ. ವೆಲ್ಕಮ್ ಮ್ಯಾಮ್.
Welcome. Thank you so much, sir. Welcome to all the students.
ಸೊ ಅದೇ ರೀತಿ ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು. ಅದೇ ರೀತಿ ನಮ್ಮ ಅಕಾಡೆಮಿ ಅಧ್ಯಕ್ಷರಾದ ಪ್ರೊಫೆಸರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ ಅವರನ್ನು ಕೂಡ ಸ್ವಾಗತಿಸುತ್ತಾ, ಹಾಗೂ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಗಳಾದಂತಹ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ ಅವರನ್ನು ಕೂಡ ಸ್ವಾಗತಿಸುತ್ತಾ, ಹಾಗೂ ಹಾಜರಿರುವ ನನ್ನ ಎಲ್ಲ ಸಹೋದ್ಯೋಗಿಗಳು, ವಿದ್ಯಾರ್ಥಿ ಮಿತ್ರರು ಹಾಗೂ ವಿವೇಕ್ ವೃಂದದವರಿಗೂ ಕೂಡ ಸ್ವಾಗತಿಸುತ್ತಾ, ಸಮಯದ ಕಾಲಾವಕಾಶ ಕಡಿಮೆ ಇರೋದ್ರಿಂದ ಸೊ ಲಕ್ಷ್ಮೀಮೂರ್ತಿ ಮೇಡಮ್ ಅವರು ನೇರವಾಗಿ ಉಪನ್ಯಾಸವನ್ನು ಶುರು ಮಾಡಬಹುದು ಮೇಡಮ್. ಓವರ್ ಟು ಯು ಮ್ಯಾಮ್.
Thank you so much, sir. Thank you. Warm welcome to all the students. So presently, the topic that has been announced to you is regulation of kidney function. Okay, so here we are not going to go into details of kidney function. I'm going to take just a few minutes to recap what the kidney actually does for you. But we are going to see that though the kidney is able to function itself in an independent manner, we still need to regulate the functioning of the kidneys, because based on everyday conditions, every minute conditions, we need to see that the body homeostasis is maintained, and therefore we have to regulate how the kidney works. And that's what we'll be learning today. So when we look at this particular organ, which is the kidney, we belong to the subphylum Vertebrata, and I'm sure the students know that subphylum Vertebrata shows the main excretory organ as a pair of kidneys, right? Now, what exactly do the kidneys do for us? Before we go to the topic, let us do a recap. So what we find in the kidneys is, kidney is called as an excretory organ, and kidney is able to supplement the work which is going to be done by the liver. So what we have is going to be this way. As most of you are aware, we find that the living cells of any organism is going to produce a particular metabolic waste product that is called as ammonia. Why exactly is this waste product produced? This waste product is made mainly whenever there is any kind of metabolic reactions involving amino acids. Then this particular waste product called as ammonia is going to be produced by living cells. Ammonia, being a highly toxic nitrogenous compound, should be immediately eliminated from the body. But to eliminate ammonia, we need to have the luxury of having a lot of fresh water available. So if there is any organism which has the luxury of having a lot of fresh water available, then it is able to send out ammonia in a soluble form from the body. And in such a case, you know, it doesn't have to worry about converting ammonia to a less toxic form. This is mainly seen in freshwater organisms like maybe mollusks, freshwater arthropods, then you have freshwater fishes, etc. They follow this mechanism where ammonia is going to be sent out in its own form. You know what is the name given to such organisms? Yes, they are called as ammonotelic forms. Some organisms will say, I do not have the luxury of having a lot of fresh water available outside me. So at the most, I can spend maybe a certain hundreds of ml of water, maybe around a liter of water if it is, let us say, a small sized dog, or it may be 1.5 liters of water if you consider human beings and gorillas, etc. So depending on the size of the organism and the kind of diet that it has, some organisms can waste a certain amount of water. So in such a case, what happens is this ammonia is going to be converted to a compound called as urea by the liver This is a particular cycle called as urea cycle, or we call it as Krebs-Henslide cycle. So with that cycle, ammonia is going to be converted to urea with the input of ATP as well as the input of carbon dioxide. And this urea is definitely very less toxic compared to ammonia. So even if it is stored in the body for some time, it is not going to produce any kind of toxic effects. And the amount of water required to eliminate this urea is going to be lesser compared to eliminating ammonia directly. So such organisms are ureotelic. Human beings are also ureotelic. Some organisms can convert ammonia to a more or less toxic form, which is going to be called as uric acid. Again, with the help of the liver itself. Uric acid is many times less toxic than urea itself. So you can imagine how less toxic it is compared to ammonia. And this uric acid, which organisms will want to do this? So in this case, the organism will say, I can waste a few ml, maybe 1 ml, maybe 3 ml, maybe 10 ml of water I can waste if I have to send out metabolic waste like that is nitrogenous waste. And they convert ammonia to uric acid. We call them as uricotelic organisms. These organisms always lay shelled eggs. It may be insects, it may be land snails, it may be your, your birds, your reptiles, egg-laying mammals, etc. We are able to do this. Right? Now, let us not worry too much about this because that was not the topic for today. Let us now consider only this part. Okay, that is living cells produce ammonia. Ammonia is converted by the liver using ATP and carbon dioxide into a compound called as urea. So, urea is made by the liver. So, if a person's liver fails, then what is going to build up in the body? Ammonia will build up in the body. Once urea is made by the liver, this urea needs to be filtered out from the blood, and that filtering out the kidney is able to do. So kidney, when we call it as the major excretory organ of the human body, what it basically does is, it is going to filter out this urea. That is going to be the function of the kidney. Now, inside the kidney, I'm sure all of you know that we have a functional and structural unit of the kidney, which is called as the nephron. Now, when you see the nephron, what is it that you're able to see? Suppose you consider a nephron which is present. We have the Bowman's capsule, and then we have the PCT, we have the leukocyte, and then we have the BCT with the collecting duct. Correct? So that is going to be your nephron structure. Till here, this is the part along with the glomerulus which actually takes care of filtering. This area is going to completely take care of filtering. So it finishes off filtering. So when I said kidneys are able to remove urea from the bloodstream by filtering it out, that is done till here itself. But I have such a large part of the nephron and the collecting duct left over. What are they going to do? They will take care of osmoregulation. That is, they will make sure that whatever vital substances are present in the glomerular filtrate can easily be reabsorbed back. Right? That is the function done by the remaining part, that is the renal tubule, as well as the collecting duct will take care of the osmoregulation part. And that osmoregulation area can itself be regulated by the rest of the body, and that is the topic for today. Okay, so the kidneys have to be regulated by the control and coordination system of the body. And that is what we are going to learn in today's topic. So it was a brief introduction to tell you what the kidney is able to do and that it needs to be regulated. So before we go to 3 different mechanisms of regulation that we will be learning today, let us understand one brief thing. Why is it that the kidney has to be regulated? Right? Now, what we find is going to be like this. Now, let us say I have back-to-back classes of maybe, 90 minutes each, and I have 3 hours of back-to-back classes that is present. Obviously, if I'm comfortably sitting at home, the amount of, exposure to sunlight, the amount of dehydration that I'm going to undergo at home is nothing compared to what I undergo in the class. Because if I'm continuously speaking, there is a lot of water that gets evaporated, right? Plus, if I'm sitting, if I'm standing for 3 hours, definitely my water intake is going to be lesser during classes compared to me sitting around, say, at home. Correct? So my water intake is less and evaporation is higher when I'm in a class. So apart from this, what is going to happen? You might realize that maybe my diet today had 3 cups of coffee. That's possible. And maybe my diet yesterday had lot of paneer items, okay, which is rich in proteins. So depending on my diet, depending on my lifestyle, I have access to water, I don't have access to water, I am sitting in a very, very humid room, I'm sitting in a very hot room which is very dry and I'm continuously sweating. So depending on the conditions, we need to see that the osmolarity of blood means how much of water is there in the blood, how much of mineral salts are there in the blood, that has to be maintained. Please remember, for the body, the quality of urine is not a big priority compared to the quality of blood. The blood has to maintain its health, its quality properly. How much of water should be there in the blood, how much of salt should be there in the blood is highest priority, and not the quality of urine. You understand that? Some person is trapped in a building after an earthquake, has no access to water at all and food. They are going to be producing very small quantity of concentrated urine compared to the same person few days ago. Why? Because the quality of blood has to be maintained properly. Electrolyte and water balance has to be maintained. And that is going to be done by regulating the functioning of the kidneys. Now, how is this regulation going to be done? Let us try and understand. Kidney function regulation is going to be more the job of the endocrine system rather than the nervous system. Does it mean that the kidneys do not have nervous input? Of course, they will have nervous input. But if you try and find out which of these control systems, on one hand nervous system, on the other hand endocrine system, you realize that endocrine system controlling the kidneys is definitely seen at a much more higher control level compared to the nervous system. And today, I am going to be explaining to all of you 3 different mechanisms that are present in order to control how the kidneys function. The first among these regulatory mechanisms is going to be called as RAS mechanism. The first mechanism is called as RAS mechanism. I will give you the full form of RAS mechanism after I finish explaining what this particular mechanism is. Right? Can we start with RAS mechanism now? Now, all of you are aware, your teachers would have definitely made you draw the nephron structure. Correct? When you draw the nephron structure, there is a tendency to draw it in this manner. That is, we draw the Bowman's capsule in this manner, and then we draw the PCT, and after that we come to the loop of Henle, and finally we are going to come to the DCT that finishes off the nephron, and then of course it will join the collecting duct. That is going to be a standard way in which we draw a nephron. Please understand that though a nephron structure is exactly present in this manner, nephrons cannot be spaced out like this. You will hardly accommodate maybe 1,000 nephrons instead of million nephrons in a kidney. So it is important that these nephrons have to be folded. Though the structure is correct, they have to be folded so that many nephrons can be accommodated in a single kidney. How will the nephrons be arranged? What I'm basically trying to tell is, in your cupboard, you will not arrange shirts without folding them. Okay, or towels or saris are not going to be arranged in a cupboard without folding them. Then we cannot accommodate many saris. So what do we generally do? We fold the sari and then we can accommodate a lot of them. In the same way, these nephrons are going to be folded over themselves in order for easy packing. How is the nephron going to be folded? Let us try and understand. So here we have this structure. This is going to be the Bowman's capsule, which is present in this manner. This is the Bowman's capsule. This is going to be the PCT, which is present here. This is the PCT, and then this is going to be the loop of Henry. I hope you are realizing that I am drawing in the same manner. I am not changing the shape of the nephron. The nephron structure is exactly the same, but I am just holding it in a better manner. And then we are going to have the DCT that is present here. That is how the DCT is going to be, which is going to join the collecting duct. That is going to be the structure for the nephron. Now, for RAS mechanism, I had to draw this diagram. Why did I have to draw this diagram? I'm sure the students know that from this side, we have the finest branch of the renal artery, which approaches the nephron. And all of you know the name of that particular branch, that is the afferent arteriole. Here I am not explaining the structure of the nephron, therefore I am not going into details. I am expecting that you know this already, so that I can stick to my topic. So this is the afferent arteriole, and then of course the afferent arteriole is going to branch off to produce a ball of capillaries, which is going to be called as the efferent arteriole. The junction between, when you look at the afferent arteriole, you will find that there is a particular place where the afferent arteriole and the DCT come close to one another. I'll repeat again. To understand RAS mechanism, we are going to the packing of the nephrons. And when packing of nephrons is done in a particular manner, you find that this is the afferent arteriole. This is going to be the glomerulus, which is present here. That is the glomerulus, which is present, which is then going to become the efferent arteriole in this manner. And when DCT comes closer to the afferent arteriole, this is DCT. When it comes closer to the afferent arteriole, at this particular junction, you will find the presence of fine cells. There are fine cells that are present at the junction of the afferent arteriole and the DCT. These cells which are present here are going to be called as the juxtaglomerular cells. These cells are called juxtaglomerular cells. Juxtaglomerular cells. That is the cells which are present at the junction of the afferent arteriole. This is the afferent arteriole. And this is going to be the DCT. At this junction, these special cells are present here, which are called as the juxtaglomerular cells, JG cells. Right? For RAS mechanism, we are going to pay attention to these cells called as the juxtaglomerular cells. This is a very important NEET question also. There are going to be some choices given where they can ask you, where exactly are the JG cells located? Is it between PCT and efferent arteriole? Is it between PCT and afferent? DCT and efferent, or DCT and afferent? And this is a good question that is asked. Another higher level question that can be asked in the NEET exam or also in the CET exam is, they can ask you regarding the nature of the JG cells. Okay, what kind of cells are they? So that also is going to be asked. So let us begin with these cells description. We will start with RAS mechanism, which is the first mechanism to regulate how the kidney functions. When exactly does RAS mechanism come into play? Let us try and understand that right now. So, whenever there is going to be a fall in blood pressure, or there is a fall in glomerular filtration rate. All of you are aware about the glomerular filtration rate, which is 185 liters per day. Okay, so when you look at the GFR or you look at a fall in blood pressure, this is going to now trigger RAS. This will trigger RAS. Let us see how this is going to be triggered. So whenever there is a fall in the blood pressure or a fall in the glomerular filtration rate, you find that RAS is going to be triggered. Who is able to detect this fall in blood pressure or the GFR? This detection is going to be done, this entire detection is done by the afferent arteriole. So the afferent arteriole which is present is the one that is going to detect that there is a fall in the blood pressure or the GFR. As soon as this detection is done, what is going to happen as a result? As a result of this, the JG cells are now going to secrete a particular hormone which is called as renin. So all the neat aspirants, please make a note here. This is not RENNIN. It is RENIN, renin. Renin is RENNIN is going to be a milk protein digesting enzyme secreted by the infant's stomach, whereas RENIN is a hormone that is secreted by the juxtaglomerular cells. So they will secrete a hormone called renin, which gives you the idea about JG cells. JG cells are endocrine in function. They are endocrine cells. Now what is going to happen next? I want you to remember that renin is going to be secreted. Let us go ahead and see what is going to happen after this. So as soon as renin is secreted, what is this renin going to do now? So the renin which is present is now going to help in conversion of a compound which is called as angiotensinogen. So it is a generator of angiotensin. It is a generator. O-gen, whenever you see that, or G-E-N, when you see, you know that it is a generator. So it is a generator of angiotensin. This compound is all the time present in the blood. Renin was not present all the time in blood. Renin got released by the kidney. That is by the JG cells, it got released and entered into the bloodstream because hormones enter into the bloodstream. This renin will now act on a compound present in the blood called angiotensinogen and help in converting it into angiotensin 1 and further into a compound called angiotensin 2. I'll repeat. So when there is a fall in the blood pressure or a fall in the glomerular filtration rate, at that point of time, you find that JG cells will release a chemical which is a hormone called as renin. Renin will help in converting a compound present in the blood called angiotensinogen and convert it into angiotensin 1 and then angiotensin 2. What are these compounds? Both the compounds are having the same function. Angiotensin 2 does it at a much more higher efficiency compared to angiotensin 1. There is no other difference between the compounds. It is just that the molecular structure is such that angiotensin 2 does the same work in a much more efficient manner. Okay, what is the function done by angiotensin 2? So angiotensin 2 now has got two roles. Let us look at the roles one by one. So the first role that angiotensin 1 has is, it is going to target the DCT. So it targets DCT. Okay, the students will very well know, because before this topic you would have done a topic called as urine formation. So you know that DCT is the only part of the nephron where conditional reabsorption of substances occur. I'm sure your teachers have explained that to you. What is meant by conditional reabsorption? PCT and loop of Henle cannot change the way they reabsorb substances. Whether I drink water, I don't drink water. Whether I drink coffee, I don't drink coffee. You will find that PCT and loop of Henle behave exactly in the same manner. Whereas the only part of the nephron which can be modified is going to be the DCT. That's why we say that conditional reabsorption occurs in the DCT. So what's going to happen? Angiotensin 2 first function is it will target the DCT. What will it ask DCT to do? It will ask the DCT to reabsorb. Reabsorb what? It will ask it to reabsorb more amount of water and more amount of NaCl. So it will ask the DCT to change its permeability slightly so that the DCT is able to reabsorb more water as well as more NaCl. What happens when you put water into the blood? It's an excellent physics question which can come even in the JEE mains, right? Because this is all to do with fluid dynamics. So what happens is, as soon as you put more water into the blood volume will increase. And when blood volume increases, blood pressure will also increase. I'll repeat again. So, angiotensin 2 first function is it will target the DCT and ask it to reabsorb more water. How does that help? So, when you reabsorb more water, automatically you find that the blood volume will increase and thereby the blood pressure will increase, which is what we wanted. Because the trigger was there was low blood pressure and no GFR. So now you have been able to increase the blood pressure, and all of you know that when blood pressure changes or increases, GFR will automatically increase. Correct? Okay. How does NaCl help in this context? So addition of more NaCl into the blood is going to definitely trigger to see that the heart rate also increases. It will also see that the blood pressure increases. That's why they say that people who are hypertensive patients, they have to cut down on their salt intake per day, right? Because we know that when you add more NaCl, naturally you find that the blood pressure is going to increase. There is a pathway for that. So with both these, what is going to happen is, you find that because of both these reasons, ultimately we find that because of this, blood pressure increases, and when blood pressure increases, GFR will automatically increase. That is going to be the function done by angiotensin 2, function number 1. Let us now look at function number 2 that angiotensin 2 does. So the second function that angiotensin 2 is going to do is, angiotensin 2 is going to now bring about constriction of, it brings about constriction of the blood vessels. It brings about constriction of the blood vessels. Whenever blood vessels undergo constriction, look at the name. Angio means vessel. Tensin means pressure. Angiotensin 2 is going to make sure that there is constriction of blood vessels. So blood vessels are going to be constricted, means made narrow. And when there is constriction of blood vessels, automatically it will increase the blood pressure. Automatically blood pressure will increase. And we know that when blood pressure increases, naturally we find that GFR is also going to increase. Now, what we have to learn is, we have to find out how is angiotensin 2 bringing about this particular function. Let me explain how it is able to bring about targeting of angiotensin II to DCT. Right? Initially, people thought that angiotensin II is directly attacking the DCT and changing its permeability. But later research showed that DCT does not have any receptors for angiotensin II. Further research said that angiotensin II is able to bring about the increase in water and NaCl not directly but in an indirect manner. And let me explain how it is able to do this in an indirect manner. So what angiotensin II does is, angiotensin II is going to target a particular endocrine gland called adrenal cortex. I'm explaining how angiotensin II can increase the reabsorption of water and NaCl. So angiotensin II will directly target the adrenal cortex. Adrenal cortex is going to be the outer part of the adrenal gland, which is an endocrine gland. When it targets the adrenal cortex, it will ask adrenal cortex to secrete a group of hormones that are called as mineralocorticoids. A group of steroidal hormones called mineralocorticoids. There are many mineralocorticoids present, but the most potent mineralocorticoid which is seen is a compound called aldosterone. Aldosterone is the most potent of all the mineralocorticoids. So what happens now? Aldosterone is the one that will target the DCT and help in the reabsorption of more water as well as more NaCl. And that is how your blood pressure and thereby the GFR will increase. Right? Now, because we are involving three major groups of chemicals, we call this as RAS mechanism. The R is going to come from renin. The A comes from angiotensin. And the AS comes from aldosterone. So RAS mechanism is renin, angiotensin, aldosterone mechanism, which is a very powerful mechanism to see that the blood pressure and thereby the GFR can increase. Before I erase the board, let me quickly explain how this mechanism is going to take place. Then we'll go to the second mechanism. So I'll repeat again. Okay, so what are we going to have like a story you should be able to remember. So RAS mechanism, when does it come into play? Whenever there is a fall in blood pressure and there is a fall which will also result in a fall in GFR, glomerular filtration rate. As soon as there is a fall in blood pressure and GFR, JG cells will be activated, which are present junction of the DCT as well as the afferent arteriole. At that junction, the JG cells are present. These JG cells will release a chemical called renin into the bloodstream. Renin is a hormone, which means JG cells are endocrine in nature. The renin will now bring about the conversion of angiotensinogen, which is a chemical already present in the blood, and converts it into angiotensin 1 and angiotensin 2. Among these, you find that the efficiency of angiotensin 2 is going to be extremely high. In the human body, there are many vasoconstrictors, that is, chemicals that can constrict blood vessels. Angiotensin 2 is considered as the most powerful one in the human body. This angiotensin 2, which is present, is now going to do two functions. What are the two functions? One is angiotensin 2 is going to constrict blood vessels, as a result of which blood pressure will increase, as a result of which GFR will increase. Angiotensin 2 will also target the adrenal cortex and make the adrenal cortex release a group of hormones called mineralocorticoids, of which the most potent one is aldosterone. Aldosterone will now target the DCT and help in reabsorbing more water. When I reabsorb more water, automatically blood volume will increase, blood pressure will increase, GFR will increase. I will reabsorb more NaCl, because of which blood pressure will increase and GFR will increase. This is called as the renin-angiotensin-aldosterone mechanism. Okay, which is one way to control the kidney function. Let us go to the second method of controlling the kidney function. So the second mechanism that I will be explaining now is called as the ANF mechanism. So the second mechanism is called as the ANF mechanism of controlling how the kidney is going to function. Okay, what is this ANF mechanism? Let me explain. So ANF mechanism is going to control the body whenever you find that the GFR and the blood pressure is high. So high blood pressure or high GFR is going to be the trigger for this mechanism to occur. So whenever blood pressure is high or GFR is high, then this mechanism comes into play. So we can also say that when RAS is happening, RAS cannot continue for a long time. You have to bring the body back to normalcy. That time also ANF mechanism is going to play a role. Okay, what is going to happen here? So whenever there is high blood pressure or high GFR content, we find this is going to be read or it is perceived by the atria of the heart. So the right atrium of the heart is able to perceive that there has been a high blood pressure. So it is able to perceive or read that there is high blood pressure. As soon as it is able to perceive that there is high blood pressure, what is going to happen at that point of time? At that point of time, this right atrium, as soon as it reads that there is high blood pressure, it is going to release or secrete a particular peptide hormone, which is called as the atrial natriuretic factor. I'll repeat. So whenever there is high blood pressure or high GFR, the right atrium is able to perceive it very easily. And the right atrium now will secrete a particular peptide hormone called as the atrial natriuretic factor or ANF. Now, what is this factor going to do? This factor is now going to target, it will target DCT. I was telling you this is where conditional control can happen. So it will target DCT. What will it ask DCT to do? It will ask DCT to do two things. Absorb less water, don't absorb more water, and absorb less NaCl. What happens when less water is reabsorbed by the DCT? You will find that blood volume will fall. Correct? You are not adding more water into the blood from the DCT. So blood volume will fall. And when blood volume falls, automatically blood pressure will get lowered. And when blood pressure gets lowered, GFR will also come down. When you reabsorb less NaCl from the DCT, naturally the blood pressure will dip. As a result, GFR will also dip. Is that clear? So the person is going to lose more water from the urine when ANF is controlling the kidneys and will also lose more NaCl from the urine. That is, through the urine, there will be a little bit more NaCl also lost by the person. And that is why you can see the name very clearly. Uritic means anything related to urine. Natrium, all of you know what natrium is. What is natrium? Natrium is the correct name for sodium. No, sodium, what do you write it as? You write it as Na, because the name for sodium is natrium. So, natriuretic, more sodium chloride in the urine. and this factor is secreted by the atria of the heart. That's why it is atrial natriuretic factor. More salt loss by the urine. This is to counteract RAS mechanism or come into play whenever blood pressure or GFR is high. If this mechanism is so good, then nobody should suffer from high blood pressure. No, not like that. It is able to control momentarily for a particular level. You understand that? Your brakes will work only when your car is running at a certain speed. You cannot expect it to work miraculously well at all speeds. That's not how it works, right? So in the same way here, this will work only for a moment, and it will work whenever the blood pressure is going to be a little bit high under controllable levels, not during all periods of time. Okay, let's go to the last mechanism. So the third mechanism that I'm going to explain today is called as the ADH mechanism. So the third mechanism is ADH mechanism. Right? So ADH mechanism, let us try and understand. So when does ADH mechanism come into play? When does RAS come into play? Whenever the blood pressure has fallen and whenever the GFR has fallen, then RAS will come into play. When does ALF mechanism come into play? Whenever blood pressure is high and GFR is high, ALF mechanism comes into play. When does ADH mechanism come into play? Whenever a person has got a loss of water from the body. So this is what is going to trigger ADH mechanism. So in what conditions can a person lose water? So let us say it is extremely hot summer and the person has been sweating a lot. That is one way in which we can lose a lot of water. Another way to lose a lot of water is if a person, let us say for some reason, has been having diarrhea. So there are loose motions, or maybe they are having bouts of vomiting. So that may also be one of the reasons why a person can lose a lot of water. So, or it may be the reverse. That is, the person has got very poor intake of water. So it need not always be loss. It can also be very poor intake of water. That also is possible. So under such a condition, ADH mechanism is going to be triggered. Now, who is going to identify that the person is almost on the verge of getting dehydrated? This is going to be detected by a number of special neurons that are present in many parts of the body called osmoreceptors. Okay, so we have osmoreceptors. They are able to measure osmolarity of blood. That is, blood has got less water. They are able to measure it very easily. These osmoreceptors will now give a feedback that there has been a loss of water or less intake of water to a part of the forebrain called hypothalamus. I repeat again, point number 1 is detected by these, and these will give a message to the hypothalamus that the point number 1 has happened. Now, what does hypothalamus do? Hypothalamus is now going to produce a hormone called as ADH. I'm sure students will know the full form of ADH. You have been learning right from 10th standard, and the full form is anti-diuretic hormone. Anti-diuretic hormone. So, if I have to learn anti, I must first know what is diuretic. Correct? Diuresis. The word diuresis means production of lot of dilute urine. So production of large quantities of dilute urine is called diuresis. I'm sure the class 12 students, second PU students, definitely would have learnt about diuretics, which are abused by sports people. You have learned it in human health and disease, right? I'm sure you know that. So diuresis is going to be production of dilute urine. Production of dilute urine, the hormone which prevents, is anti-diuretic hormone. So hypothalamus will produce ADH and send it via the neurons to a particular part of the pituitary gland called as neurohypophysis. Neurohypophysis. We know it by its common name, posterior pituitary or pars nervosa. I'll repeat. So the first point is detected by the osmoreceptors in the body. They will send a message to a part in the forebrain called hypothalamus. Hypothalamus will secrete or will manufacture, rather, manufacture is a better word, manufacture a hormone called ADH. ADH is then sent via neurons to the neurohypophysis, which is the posterior pituitary gland. And this is now going to release ADH. So, posterior pituitary doesn't manufacture ADH, it will only release it down. So, ADH release is going to happen. What is this ADH going to do? Let us try and understand. So, ADH has got two functions. What are the functions that ADH should do? Please remember what has to be reversed. This has to be reversed, or this has to be tackled rather. So what are the two things that ADH will do? Point number one, what it is able to do is, it will now target the DCT. I've repeated so many times, this is the one that can undergo conditional modification or conditional reabsorption. So ADH will target DCT and it will ask DCT to reabsorb more water and more NaCl. Isn't it that I had problems with water loss? Correct. Nobody loses pure water. If I produce, if I undergo vomiting or I undergo diarrhea, etc., I will be losing a vital mineral salt, which is NaCl. So NaCl loss is also something that is usually seen along with the loss of water. Even when you sweat a lot in hot summer, you are having a lot of loss of water. So there also NaCl is going to be lost. So more reabsorption of water and NaCl will happen. When you reabsorb more water, naturally you find that the blood quality is going to improve and your dehydration will come down. What else does ADH do? So ADH will also target the blood vessels of the body. So it targets the blood vessels of the body and will constrict them. It will make them narrow. So when blood vessels of the body become narrow, what happens is blood pressure will increase, the GFR will increase, more chance for the DCT to reabsorb water and NaCl. Because if you send blood to the sweat glands, you can't control the sweat glands. But if you divert more blood towards the kidneys, you have more chance at reabsorption. So you will be constricting the blood vessels to see that GFR increases and there is more chance at reabsorption of water and NaCl. Therefore, because blood vessels are undergoing constriction, we call ADH by its old name, and it is also called as vasopressin. Vasopressin is the old name. Vaso refers to vessel, pressin means to constrict. So that is going to be ADH. So as a result, this problem is going to be sorted out. So whenever a person, many a times we see when students are writing the exams, okay, like JEE or KCET or NEET, etc., they don't drink enough of water. And that's why they'll have lapse of memory. So they have to continue to drink enough of water. And suppose for some reason somebody is not being able to drink enough of water, they are under the danger of dehydration. That is when the osmoreceptors will ultimately help in the give the message that ADH has to be released. ADH will target the DCT. It will also target the blood vessels and will help in reabsorbing more water and NaCl. It will also constrict the blood vessels, so there is more chance for the DCT to reabsorb water and NaCl. These are the three mechanisms which are in play for the regulation of kidney function. Is that clear? All the three mechanisms. And as I said, even for physics, you can have questions coming from here, and the questions are going to be pertaining to the fluid dynamics of blood. How more water or less water can affect even the pressure of blood. So such questions. So even for non-biology students, such a biology-based question can be asked in physics many times. Right? So three mechanisms. I'll write down all the three mechanisms names for you on the board. We learned first RAS mechanism, that is renin, angiotensin aldosterone mechanism, which is triggered when there is fall in blood pressure and fall in GFR, and through three different hormones or three different chemicals, let us say. They are two lesser known to be called as hormones as such. Definitely they are hormones. That is renin, angiotensin and aldosterone. You are able to increase blood pressure and GFR. And the second mechanism that we learned was ANF mechanism, where the person is going to lose more water and NaCl through the urine. Natriuretic factor mechanism. And the third mechanism was the ADH mechanism, which comes into play whenever the person loses more amount of water. Right? Yes, sir. Over to you, sir, so that, Gauda sir, so that we can go ahead if there are any questions as such.
Students, please ask any questions and any doubts on today's topic.
Or you can send me the questions if they have any doubts.
Later on, I send ma'am chat box.
No problem. Sure, sir. Sure.
ಜಾಸ್ತಿ ವೆಬೆಕ್ಸ್ ಅಲ್ಲಿ ಇವತ್ತು ಅಟೆಂಡ್ ಆಗಿಲ್ಲ ಮೇಡಮ್, ಜಾಸ್ತಿ ಒಂದು 20% ಆಗಿದ್ದಾರೆ ಅಷ್ಟೇ.
Okay.
ಯೂಟ್ಯೂಬ್ ಅಲ್ಲಿ ಜಾಸ್ತಿ ಜಾಯ್ನ್ ಆಗಿದ್ದಾರೆ.
Okay. Right. Yes.
ಹಾಗಾಗಿ ನಾನು ಲೇಟರ್ ಆನ್ ಐ ಸೆಂಡ್ ಥ್ರೂ ಮೇಲ್ ಮ್ಯಾಮ್.
Sure, sir. No problem. Yes.
ಸೊ ಥ್ಯಾಂಕ್ ಯು ಮ್ಯಾಮ್. ಇವತ್ತು ತಾವು ಕೊಟ್ಟಂತ ಉಪನ್ಯಾಸ ಕಿಡ್ನಿ ಕಾರ್ಯನಿರ್ವಹಣೆ ಮತ್ತು ಅದರ ತೊಂದರೆಗಳು ಮತ್ತು ಅದರ ಪರಿಹಾರವೇನೆಂಬುದರ ಮೇಲೆ ಬಹಳ ಸರಳವಾಗಿ ಕನ್ನಡ ಮತ್ತು ಇಂಗ್ಲಿಷ್ ಎರಡರಲ್ಲೂ ಕೂಡ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ಅನುಕೂಲವಾಗುವ ರೀತಿಯಲ್ಲಿ ಬಹಳ ಪೂರಕವಾಗಿ ಹಲವಾರು ಮಾಹಿತಿಗಳನ್ನ ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ತಾವು ತಿಳಿಸ್ಕೊಟ್ಟಿದ್ದೀರಾ ಮೇಡಮ್. ಸೊ ನಮ್ಮ KST ಅಕಾಡೆಮಿ ಪರವಾಗಿ ಹಾಗೂ ನನ್ನ ವೈಯಕ್ತಿಕವಾಗಿ ಧನ್ಯವಾದಗಳು ಮೇಡಮ್.
Thank you so much.
ಅದೇ ರೀತಿ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಅಧ್ಯಕ್ಷರಾದಂತ ಪ್ರೊಫೆಸರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳು ಸರ್. ಹಾಗೂ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನು ಆಯೋಜಿಸಿ ನಮಗೆ ಪ್ರತಿನಿತ್ಯವೂ ಕೂಡ ಸಲಹೆ ಸೂಚನೆ ನೀಡುತ್ತಾ ಬಂದಿರುವ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಗಳಾದಂತಹ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳು ಸರ್. ಸೊ ಅದೇ ರೀತಿ ಈ ಕಾರ್ಯಕ್ರಮವನ್ನು ವೀಕ್ಷಿಸಿದಂತಹ ಹಾಗೂ ನನ್ನ ಸಹೋದ್ಯೋಗಿಗಳು, ವಿದ್ಯಾರ್ಥಿ ವೃಂದದವರು ಹಾಗೂ ವಿವೇಕ್ಸ್ ವೃಂದದವರಿಗೂ ಕೂಡ ನನ್ನ ಧನ್ಯವಾದಗಳನ್ನು KST ಹಾಗೂ ವೈಯಕ್ತಿಕವಾಗಿ ತಿಳಿಸುತ್ತಾ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನು ಮುಗಿಸುತ್ತಿದ್ದೇವೆ. Once again, thank you, ma'am.
Thank you so much, sir. And in case the students have any, particular topic of preference, or board exam ella barta ide, yav idre please reach out to me. I will try to do my best.
Yes, sure, ma'am. I will send.
Thank you, sir.
ಮತ್ತೆ ಇದಕ್ಕೆ ಸಂಬಂಧಪಟ್ಟಂಗೆ ಒಂದು ನೀವು ಫ್ರೀ ಆಗಿದ್ದಾಗ ಒಂದು ನೋಟ್ಸ್ ನಮ್ಮ KST ವೆಬಿನಾರ್.
Yes, sir. Definitely. Yes, sure.
End of this session and end of this program.
Yes.
ಒಂದು ಕಿರುವತ್ತಿಗೆ ರೂಪದಲ್ಲಿ ನಾವು ಒಂದು ಪುಸ್ತಕನ ಹೊರತರ್ತಾ ಇದ್ದೀವಿ.
Sure. We'll do that. Yes.
Okay. Anyway, once again, thank you, ma'am.
Thank you so much.